Hello everyone, welcome to my channel BioFM. Today our topic is muscle contraction. It is initiated by a signal sent by the central nervous system via a motor neuron. The mechanism of muscle contraction is best explained by sliding filamentous hypothesis. According to sliding filamentous theory, contractions of muscle fiber occurs by the sliding of actin over the myosin. The steps are: Step 1. An impulse from the CNS reaches the neuromuscular junction via motor neuron. Step 2. A synaptic vesicle releases neurotransmitter acetylcholine. It generates action potential in the sarcolemma that spreads through the muscle fiber. It causes release of calcium ion from sarcoplasmic cisternae into sarcoplasm. Step 3. When a muscle is relaxed, myosin heads are unable to bind to actin. This is because the active sites for the myosin heads on the actins are blocked by tropomyosin. Thus, myosins are unable to bind with actin. Tropomyosin is held in place by troponin. Step 4. When the concentration of calcium is raised in the sarcoplasm, calcium binds to troponin. This causes the conformational changes in troponin. This causes troponin-tropomyosin complex to be shipped away from the active sites. And ATP molecules attach to the active site of the myosin head. The head acts as ATPase enzyme. This causes hydrolysis of the ATP in presence of calcium and magnesium to produce ADP and PI. The myosin heads takes up the energy release and become highly energized. Step 5. Using energy from ATP hydrolysis, myosin heads binds to active site on the actin to form crossbridge. Step 6. This pulls actin filament on both sides towards the center of A bands. Actin filaments primarily overlap so that H zone disappears. The Z lines attached to actin is also pulled inward. It causes a shortening of sarcomere. I bands get shortened, whereas A bands retain the length. Myosin releases ADP and PI and goes back to its relaxed state. A new ATP binds and crossbridge is broken. The ATP is again hydrolyzed by the myosin head and the above process is repeated, causing further sliding. When calcium ions are pumped back to sarcoplasmic cisternae, actin filaments are again masked. As a result, Z line returns to its original position. It results in relaxation.